Today, what I'd like to do is talk about biomarkers of effect, and specifically defining the relationship of safety biomarkers and disease biomarkers with respect to biomarker qualification. As you know, this is a part of the workshop of biomarkers of effect, safety biomarkers as disease biomarkers. We're going to have a panel session on November 3rd. I'm going to be a part of it, and I'll introduce the panel in my last slide. But let's go ahead and talk a little bit about biomarkers of effect. This was a phrase that I was introduced to a few years ago. I was giving a presentation for an environmental toxicology group, and they brought up this phase, and I defined it, this phrase, and I define it here on the slide. It's defined as any measurable biochemical, physiological, or other alteration within an organism, depending on the magnitude, that can be recognized as an established or potential health impairment or disease. And I put the references of where this came from in the 1980s and in the early 2000s. Now, the part that was surprising to me is this is very, very different than the way we think about biomarkers within drug development. Instead, what we've done in the drug development realm is to go ahead and sequence or separate the various biomarker types so that, as we work towards having these biomarkers to be used within drug development, we know exactly what we're trying to, chase after as far as the different properties and characteristics of the biomarkers. And so, as many of you know, the Predictive Safety Testing Consortia is very much interested in safety biomarkers. And these are biomarkers that basically will tell us whether or not, a toxicity or an adverse effect is happening or going to happen with drug exposure. However, we have a whole litany of disease or efficacy biomarkers that are also subcategorized, right? Diagnostic, monitoring biomarkers, prognostic biomarkers, pharmacodynamic and response biomarkers. And what I've done here is I've taken the definitions from the best resource that the FDA and NIH put together, several years ago. I think these are really good because they do allow us to have a discussion about what the biomarker is about. But what they don't do is they don't allow us, like that definition for biomarkers of effect, to go ahead and commingle the biomarkers, right? And understand how they can be used differently within drug development, even though it's the same biomarker. Let me go ahead and go a little bit deeper into this. So, if we go ahead and we look at how we think about biomarkers of drug development, we've separated, safety biomarkers from disease and efficacy biomarkers. But ironically, many of the safety biomarkers that are used within drug development actually are derived from safety biomarkers used in biomarkers used in clinical practice to go ahead and diagnose diseases in patients. I mean, if you go ahead and look at the biomarkers, the clinical chemistry that your physician gives you at your yearly checkup, well, they're pretty darn close to the battery of safety biomarkers that are used in drug development. Many of these biomarkers are actually biomarkers of organ injury or organ function. And so I think what we're finding, in doing this very simple analysis is that in reality, these safety biomarkers and biomarkers of disease detection, diagnostic biomarkers, or even those used in efficacy have significant overlap, right? They're one and the same. And so we can almost say that nearly all safety biomarkers can be used as disease or efficacy biomarkers. There's no doubt that there's going to be some outliers on both sides. And these are going to be biomarkers that are primarily driven by the mechanism of the drug. We had a really great discussion within the the working group that pulled this webinar together. And, we basically posed that question is, what are those outliers? Are there actually outliers? And the only time that we could come up with outliers is when a biomarker is very, very specific to either a disease's or a drug's mechanism of action or its mechanism of toxicity. So something to think about. Could be something we could talk about within the panel session. Again, I challenge you, tell me I'm wrong here. I'd love to hear it. So let's say I'm not wrong, right? And let's say that this statement is true, right? That nearly all drug safety biomarkers can be used as disease or efficacy biomarkers in drug development. That would then mean that it's going to be easier to take qualified biomarkers and actually move them to the other class. In other words, safety biomarkers being used within the disease or efficacy space, or disease and efficacy space biomarkers being used in the safety space, right? We should be able to leverage the data that's generated around these biomarkers in order to accelerate their qualification within the other arena. So guess what? We already do this, right? This has already been done. This isn't something that's new, right? So, automatically when I began thinking about, okay, what can I think about for a biomarker that has overlap in both safety as well as in disease or efficacy, alanine aminotransferase came to mind. And this is a biomarker for hepatocellular injury, right? So drugs that damage the hepatocytes in the liver, ALT goes up. Diseases that damage the liver, ALT goes up because there's hepatocellular death going on. And what we find in two separate FDA guidances, it speaks to how ALT can be used as either a disease diagnostic or even as a pharmacodynamic endpoint in a study. Well, in another guidance, we find that guidance speaking to us how to use ALT for detecting drug-induced liver injury. So that overlap is already there, right? And that thinking is already embedded with health authorities. Likewise, if we look at some of the qualifications that the PSTC is currently facilitating, that around glutamate dehydrogenase, as well as a panel of skeletal muscle injury biomarkers, both the FDA and the EMA have allowed us to use samples from patients with specific organ diseases to support the safety biomarker qualification. Now, right there, that speaks to the fact that the basic mechanism by which disease or drug-induced injury is eliciting these biomarkers or causing these biomarkers to go up in biological fluid is similar or the same. And I think that's what then joins safety biomarkers with disease and efficacy biomarkers. So let's go ahead then and think about how we can leverage the data that's produced by any type of qualification project, right? We can start off with basic biomarker discovery, where we begin to understand the basic science behind the biomarker and why we think it's going to be a good biomarker for a given approach, right? All that data is transferable no matter how that biomarker is being used in disease or in safety. Likewise, how we measure the biomarker, the assay, the measurement technique, likewise for fluid as well as imaging biomarkers, is probably transferable, at least in part. It may need to be further updated for how we're going to use that biomarker, but we can talk about that in a second. Now, the clinical validation is probably going to be completely different. It's going to be unique between safety and disease because we're using a different patient population, and we're going to have to go ahead and understand how that biomarker is behaving in those two patient populations. Even with safety biomarkers, we have to do this work, right? If we're looking at normal healthy volunteers, that's one study population where we want to use that safety biomarkers. But now if we move to patients with a disease, there we're going to have to understand how that disease population is going to respond according to that safety biomarker. So, again, conceptually not different thinking from what we've been talking about with biomarker qualification in general. So let's talk about biomarker qualification. For many of you, you've seen this slide, right? Ad nauseum, right? Many, many times. But for a couple of folks in the audience, this may be new for you. And this is on the bottom, what we have is the process by which the FDA works with requesters to go ahead and have a biomarker thought, if you will, or approach presented to the FDA all the way through to what data is required and what that final qualification package looks like. The conceptual process is on top, right? Where we think about, okay, so what's the drug development need? What are we trying to do? We create a context of use, how we're going to use the biomarker. There's a certain level of evidence that's required for us to achieve qualification. And then hopefully we do achieve qualification and we have an endorsed drug development tool or solution that can then be used by drug developers to accelerate their given project around a given clinical candidate. Now, if we think about this again, this process, both the conceptual as well as the operational, could be accelerated if indeed we're looking at a second qualification, because a lot of the legwork and characterization of the biomarker would have already been done. So if we divide up qualification really into three buckets, right? First, the context of use. Second, the analytical validation. And then third, the clinical validation. I think what we can do is dissect and understand where we need to add additional data if we're moving from the safety space to the disease or efficacy space. And then I think that's helped drawn out here, where we define really what the context of use is. And that's a description of what the biomarker is going to be used for and how the biomarker is going to be applied in drug development, right? It's going to dictate the species that it's going to be in, the population, etc. But this context of use is really, really important because it actually defines then what the analytical validation as well as the clinical validation needs to be. And it also will help you then define the relationship between the two, right? And let me go a little bit further in the detail on that. And between the two, I mean the assay and the analytical validation, or the assay and the clinical validation. So here is a more granular look at how with safety biomarkers, we go ahead and qualify these biomarkers, right? At least from a clinical validation standpoint. We go ahead and use non-clinical species to go ahead and use prototypical toxicants to demonstrate end organ toxicity. We compare the response of the novel biomarker to standard biomarkers, as well as the truth. And the truth here is histopathology. Hopefully what we find is that this novel biomarker performs in a superior fashion to that of the current standard. We would then take that biomarker into humans and go ahead and compare that novel biomarker again to the current standard biomarkers under conditions where we're actually seeing organ injury. Now, that could be where a toxicant was given and we're seeing organ injury in a human, or it could be under the conditions where we're actually seeing a disease, right? And that can help us then understand the responsiveness of that biomarker so that we can then hopefully move forward and begin to utilize that biomarker. So We also have a pretty granular approach to how we're looking at assay validation, and indeed, it has a life cycle, right? What we find is that very early on, when we're characterizing the biomarker, is that the assay performance is pretty poor, right? Because we really don't know what type of performance characteristics we're shooting towards. But as we work through the clinical validation of the biomarker, we begin to understand different attributes of the biomarker, its baseline levels, how it responds to a toxic insult, inter and intra-subject variability. And then we also begin to understand things around the assay, such as how the samples need to be prepared or treated pre-analytically, whether the biomarker is stable or not in the given fluid that we're looking at. And so all this data really helps us then optimize that assay, right? And then hopefully at the end, what we'll be able to do is to be able to come up with a fit for purpose assay that's able to analyze that biomarker for that given context of use, and importantly, the group that we're trying to actually measure the biomarker in, right? So its use, the specific patient population, and then how that biomarker is going to be actually applied clinically. And so what this creates is a really intimate relationship between the analytical validation and the clinical validation. And that's important because if we're going to change how we're going to use the biomarker, well, we're going to have to optimize some of these factors. And I think that intimate relationship is shown very nicely here on slide 13, where we can see that as we learn about the way the biomarker is behaving in the clinic, we can optimize the assay. So in the end, we have both a fit for purpose analytical validation as well as a fit for purpose clinical validation. And I think this is really the key to any type of biomarker, either qualification or even use, right? These are the factors that you need to understand. But remember, they all relate back to the context of use, how you want to utilize the biomarker in a given patient population for a given outcome in a clinical trial. So how can we wrap this all up? It's actually pretty easy to wrap up. I mean, I think with what you've seen today, hopefully, and what you're going to see in the other presentations that are associated with this webinar, is that, if we qualify a biomarker for a safety indication, we can more rapidly move towards either a disease detection or a drug efficacy response assessment biomarker by leveraging our pre-existing biological and analytical data associated with the biomarker. However, like I was talking about just a second ago, you're going to have to be careful because both that analytical and clinical validation is, again, fit for purpose. And so since we're proposing a new use for the biomarker, we're going to have to think about how we need to modify those data sets and build new data sets, right? In some cases, it is just simply going to be, a characterization of the way the biomarker is performing, in that new patient population. Well, in other cases, a given context of use may drive you to actually reevaluating the entire analytical or clinical package. Now, again, I think it's really, really important, this last point, to remember, this isn't a new concept, right? We're just trying to expand the thinking of what we can do with our safety biomarkers. We do a ton of work around those biomarkers. And so how else can we use them? Health authorities have already basically accepted this idea from the standpoint of these commonly used biomarkers like ALT. Likewise, we see that that health authorities allow us to use clinical samples from disease populations to demonstrate how our safety biomarkers are working. And so I think we're, for the most part, there in this conversation. But I think during our panel session, we should reinforce that and get some additional feedback from some of our regulatory participants. So what we're also going to hear in the other lectures within the webinar are from Yuri Albrecht, Tanya Zapka, and from Gary Friedman around how safety biomarkers can be applied as disease biomarkers or efficacy biomarkers. Yuri is going to be talking about GLDH. Tanya will speak to the skeletal muscle biomarkers, and Gary Friedman will speak to our kidney injury biomarkers. These are all projects within the PSTC that are moving towards, safety biomarker qualification. But, like good scientists, we're always thinking about what comes next. So hopefully everyone will be able to join us on November 3rd, where we're going to have a panel discussion around the various presentations that were given. You can see the list of panelists here. We have individuals from various pharmaceutical industry groups, as well as from folks from the US FDA. Nick King, Executive Director of the Predictive Safety Testing Consortia, will be moderating the panel session. So I'd like to thank everyone. And if indeed you do have any questions, make sure you bring them to the panel. Thank you.